topic I will discuss is about phylum Acanthocephala. For introduction, Acanthocephalans are obligate parasite, which means that they are dependent on their host and cannot live without the host, and possess an indirect life cycle, which involves a vertebrate as the definitive host and an arthropod as the intermediate host. They are also known as spiny-headed or thorny-headed worms. They are also necrotrophic worms that live as adults exclusively in the vertebrate small intestine. Necrotrophic parasite means that they derive their nutrients from dead host cells that are usually killed by the parasite. Furthermore, these parasites have evolved a gruesome strategy for host parasitism known as brainjacking. Brainjacking means that they are able to alter the behavior of their intermediate host so as to increase the likelihood that the acanthocephalan will be delivered to its final host. In many cases, the parasite provokes the intermediate host to move to a vulnerable location where the final host can eat them. They are characterized by an invertible proboscis as the attachment organ, sexual dimorphism, males with semen glands, and a uterine ball in females. Acanthocephalan use the retractable and invertible proboscis to attach to the intestine of their host. The proboscis-bearing hooks and spines vary greatly among species in size and shape. Here in the figure, it shows the varying length of the proboscis on different species. In the first, it is the proboscis of the Centrorhynchus robustus. The second is the Oligocanthorhynchus tertosa. The third is the Polymorphus cuculatus. In the fourth, the Metriorhynchus centaurorum. And the last is the Plagiorhynchus cylindrius. Spine or thorns may also occur in the body. Acanthocephalans also vary greatly in size and appearance. They may be less than 1 millimeter long to over 1 meter and may be smooth or wrinkled. The phylum Acanthocephala comprises more than 1,100 valid species. Bony fishes are the most exploited group of vertebrate acanthocephalan hosts, followed by birds, mammals, amphibians, and reptiles. Here in the figure, for figure A, a young female Leptorhynchoid is Decatus. It is female because it has uterine bell, while figure B is a male Leptorhynchoid is Thecatus, a young male wherein you can spot the semen glands and other male reproductive organs. For the life cycle, the vertebrate hosts usually become infected by ingesting an infective larva called a cystecant, contained within an infected invertebrate intermediate host. Within the intestine, the diuresis worms mature, mate, and upon patency, ova containing the embryo known as an acanthor are passed in the host feces. The intermediate hosts become infected by the ingesting ova in fecally contaminated soil, food, water, or by feeding directly on the feces. The ovum hatches within the intestine of the arthropod intermediate host, releasing the acanthor that uses spines to penetrate the intestine. Within the body cavity of the arthropod, the worm develops into the infective cystecant. Natural definitive hosts includes rats, swines, and raccoons. The insect intermediate hosts vary by species, but it is usually scaraboid or hydrophilid beetles or cockroaches. In human, they became aberrant parasites which infect the human host, but they cannot develop further. However, in some acanthocephalan life cycles, another vertebrate host is utilized between the arthropod intermediate host and the vertebrate definitive host. In such hosts, known as paratenic host, the worm penetrates the intestinal wall and localizes in the mesenteries or viscera, where it remains in the infective cystecant stage. Although paratenic hosts have not been demonstrated to be physiologically required by the worms to attain maturity, they may be required to complete transfer of worms from intermediate host to the trophic level at which the final host feeds. For instance, in the figure, frogs, snakes, and salamander facilitate the transfer of worms from an arthropod intermediate host to the birds of prey that serve as the final host. For its taxonomy, phylum Acanthocephala has four classes, namely class Archaeacanthocephala, class Polyacanthocephala, class Eucanthocephala, and class Phalacanthocephala. For class Archaeanthocephala, it has four orders, namely Aporo, Rhynchida, Gigantorhynchida, Moniliformida, and Oligocanthorhynchida. For class Phalacanthocephala, there are three orders, namely Echinorinchida, Heteromorphida, and Polymorphida. For class Eucanthocephala, there are two orders, namely Geracantocephala, Neoechinorinchida. For class Polyacanthocephala, there is only one order, namely Polyacantorhynchida. For class Archaeacanthocephala, the defining morphological character under this class is that their proboscis hooks are in spiral, which is shown in the figure E, and number 4 in the figure F. A single ligament sac in the females, which is shown at the figure F number 3, and 8 semen glands in the male, shown in figure F number 2. This number 1 is the egg, and the 5 is the female reproductive system. For class Polyacanthocephala, the defining morphological characters under the class Polyacanthocephala are the two distinct ligament sacs in the females, and two elongate firiform to tubular cement glands with giant nuclei in the males, and the proboscis is long and enlarged in the apex and constricted at the base, which is shown in the figure. For class Eucanthocephala, the defining morphological characters under the class is the two ligament sacs in the females and a single cement glands in the males. Here in the figure, in number 32, it is the male with cement gland with 12 giant nuclei. In 33, the gravid female with two ligament sac. In 34, the reproductive system of mature adult male. In 35, the proboscis hooks of the male. In 36, the egg. And 37, the proboscis and receptacle of the female. And in 38, the reproductive system of the female. In class Phalacanthocephala, the defining morphological characters under the class Phalacanthocephala is that the males have usually 2 to 7 cement glands. The ligament sac in the female breaks down so that the eggs develop in the body cavity, and the proboscis hooks occurring long rows and spines are present on the body of some species. In the figure, in number 1, it is a male, showing the anteriorly enlarged proboscis, the distribution of trunk spine within the range of the long receptacle, and the posterior distribution of the reproductive system. In number 2, a posterior ventral trunk spine of a female specimen. In 3, detailed male reproductive system. In number 4, a ripe egg. And number 5 is the detail of the uterine bell of the female specimen. In number 6 and 7 are the dorsal and the ventral hooks at the mid-proboscis of a female specimen. In number 8, the detail of the vagina. In number 9, a complete female reproductive system characterized by the long and wide uterus. Acanthocephalosis. Acanthocephalosis is a disease caused by the intestinal helminth parasites of the phylum Acanthocephala. The acanthocephalans parasitize a wide variety of the terrestrial and aquatic vertebrates, both marine and inland, are found in the intestinal tract of amphibians, reptiles, birds, swine, rats, and humans. However, more than half of the known species are endoparasites of wild and farm fish. The consequences of acanthocephalan induce reaction and energy efficiency and altered metabolisms of hosts are likely to be focused more sharply with increasing emphasis on aquaculture. Shortly after infection, the proboscis is surrounded by necrotic tissue, become hemorrhagic, and inflamed after a few days. During the second week after infection by species that penetrate deeply, inflamed tissue around the anterior portion of the worm is dominated by monocytes and macrophages, maturing into epithelioid cells. An outer belt of connective tissue appears. This chronic stage often results in a fibrous nodule visible on the outer surface of the intestine. Species that do not penetrate deeply move about in the intestine and change their point of attachment before the connective tissue response result in nodules. Here in figure K, it is a photograph of Acanthocephalus dirus attached to the prolapsed rectum of a mottled sculpin. In figure L, it is the external view of the intestine from a quillback, showing a parasite-induced nodule, an expansion of the intestine compared with a normal gut. In figure M, it is the transverse section through the region of the pyloric caica of a green sunfish, infected with Leptorhynchoides decutus, showing a paradise cecum at the top and the enlarged parasitized cecum at the bottom. For figure N, the section of CHOP or Leuciscocephalus intestine showing penetration of Pomporhynchus levis. Note the hyperplasia of the lamina propria, the arrow, around the neck, and the bulbous proboscis, which is the P of the worm, forming a nodule in the caudal